The overall goal of our easy-to-implement procedure is to demonstrate fast, detergent-free delivery of membrane proteins into lipid bilayers of various sizes, using fusion between lipid bilayers formed of oppositely charged lipids.
Our methodology can be used in biochemistry and biophysics for one-step assembly of biomimetic, multi-component lipid bilayer systems from their individual components.
It is important to mention that the advantage of our approach is an easy and fast functionalization of namely complex monolipid bilayers with proteins, lipids, and other molecules of interest.
This methodology can also be used to study membrane transport processes that require energization of the lipid membrane. For example, primary pumps can be used to drive secondary transport enabled by other membrane proteins.
Our method can also serve the needs of synthetic biology. For example, to create membrane vesicles with various programmed functionalities. In future, trapping small vesicles like these inside similar larger ones may be a step towards construction of an artificial cell.
To form the lipid and oil solution, mix chloroform stock with 1 milliliter of hexadecane in a 1.5 milliliter microcentrifuge tube. Next, to evaporate the chloroform, constantly mix and heat the mixture in a horizontal shaker at 80 degrees Celsius for 30 minutes by leaving the tube open. After evaporation, close the tube and cool down to room temperature. Next, to form the lipid monolayer at the lipid-in-oil aqueous buffer interface, take a 1.5 milliliter centrifuge tube and transfer 200 microliters of lipid and oil solution